Hello to the virtual attendees of AOA 2025, and welcome to the digital spiel for this poster. My name is Lynn Kurtaf, and I am a postdoc cognitive science researcher in Gregory Hickok's lab at UC Irvine, and I'm also an SLP. I am also the lead on this project, so hopefully I can do it justice in my spiel today. Our lab at UC Irvine is connected to CSTAR, the Center for the Study of Aphasia Recovery at the University of South Carolina. This project is really a marriage of the CSTAR work and the more foundational theoretical work that Dr. Hickok and his lab are known for. A couple of years ago, Dr. Hickok and colleagues put out a paper provocatively titled Beyond Broca. The goal of this manuscript was to introduce a new model of motor speech coordination, because historical models of speech production have a tendency to underspecify the exact stages of what happens beyond phonological retrieval and articulation. This figure is a little intimidating. It has a lot going on, but I want to focus on two aspects of this model as they pertain to the results I am presenting in this poster. First, these solid green and purple areas are the two precentral speech areas, as Hickok and colleagues put it. These two nodes on the precentral gyrus are the termini of the two theorized motor speech coordination pathways. The green dorsal node is, aptly put, the dorsal precentral speech area. This area of the brain partially overlaps with language areas in the middle frontal gyrus and laryngeal motor cortex in the central sulcus and surrounding tissue. The DPCSA pathway governs the laryngeal aspects of speech, which can be summarized as prosody, speech rate, breath control, pitch, etc. The purple ventral node is the ventral precentral speech area, and it overlaps with orofacial motor cortex and the posterior aspect of Broca's area. The VPCSA pathway governs the supralaryngeal aspects of speech, meaning articulator movement and coordination. The second part of this figure I want to highlight is the connections of the DPCSA and the VPCSA. The DPCSA is connected to auditory cortex, while the VPCSA is connected to somatosensory cortex in the inferior parietal lobule. This is maybe spoiling my results a little bit, but I think these connections are very important. So that's the theoretical side of things. The clinical side of things is how this model may have explanatory power for apraxia of speech. If you're attending AOA, then you are probably aware that apraxia of speech can be difficult to diagnose and treat, and there are a lot of debates surrounding the disorder. Recently, one of the larger primary progressive apraxia of speech labs at the Mayo Clinic proposed a classification system that would split PPAOS into prosodic and phonetic subtypes. Sound familiar? Prosodic PPAOS is characterized as a difficulty with syllable segmentation, speech rate, and breath control. Phonetic PPAOS is characterized as the presence of sound distortions and sound substitutions. Because C-star has a large stroke cohort, the question I am interested in answering is whether or not post-stroke AOS, in contrast to the progressive AOS in which these subtypes were originally described, follows a prosodic and phonetic split in terms of the neuroanatomy of the disorder. This could also potentially provide some validation for the dual motor coordination model if these subtypes map onto the dorsal and ventral motor coordination pathways proposed in Beyond Broca. To accomplish this, we used the Predicting Outcomes of Language Rehabilitation, or POLAR, dataset from CSTAR. These data are approximately 120, or 100 after my exclusion criteria for this study, individuals with post-stroke aphasia. A really interesting component of this dataset for studying apraxia of speech is that a large portion of these patients also have AOS, almost 50%. AOS is quantified in these data using the Apraxia of Speech Rating Scale, which was developed by that same Mayo Clinic group studying PPAOS. The ASRS is conveniently split into prosodic and phonetic subscores, so those are the behavioral measures we chose to map onto patterns of brain lesions. Here is some demographic information as well as a visualization of lesion coverage. Pause the video now if you'd like to review any of the details. One thing I would like to point out for anybody interested in lateralization is that this study only recruited left hemisphere stroke survivors, but we've submitted a grant to collect data from right hemisphere stroke survivors moving forward, so maybe reach out in a few years. On to the methods of this project. The really short summary is that this is lesion symptom mapping. However, this is a new technique that we developed in-house in Dr. Hickok's lab in collaboration with Grant Walker, a project scientist, and Akbar Hussain, a PhD student. The way the data are interpreted is slightly different than conventional voxel-based LSM or even SVR-LSM if you are familiar with those methods. The driving ethos of this technique was to find a way to map behavioral patterns onto the brain in a more probabilistic way, ideally minimizing researcher degrees of freedom along the way to help interpretability and reduce opportunity for p-hacking. The core question we are asking is whether there is a combination of brain regions that, when treated differently from the rest of the lesion volume, increase our ability to predict behavioral scores on held-out data. The set of regions that increases our predictive power is then important in supporting the behavioral function of interest we're modeling, in this case, prosodic and phonetic subscores of the ASRS. The first step of this technique is to use permutation testing to identify individual brain regions in a given atlas that significantly model behavior. This is done using a simple multivariate linear regression, where the predictors are lesion volume within the region of interest and lesion volume outside the region of interest. Any region below the specified p-value threshold is classified as a candidate region. The second step is to calculate prediction error for every possible combination of candidate regions via cross-validation. Those errors are then compared to a control model, a univariate model that uses total lesion volume to predict behavioral scores. So, if a combination of candidate regions significantly outperforms the control model, that means treating that combination of regions differently improved your ability to predict behavioral scores. We note each combination of regions that does significantly outperform the control, and then we combine them in terms of their relative frequency to get what we call the ensemble network. The perk of looking at brain behavior relationships like this is that we aren't proposing a single solution of brain regions that supports a behavior. Instead, we are giving a probabilistic estimate of each region's contribution to modeling that behavior. However, most LSM methods people are familiar with do provide a single solution, so we can use the single best combination of regions in the ensemble network, which is quantified by minimal prediction error, and treat that as our singular solution. This is what we refer to as the critical network and should facilitate interpretation for those familiar with conventional LSM techniques. Now that we've walked through the methods, let's look at the maps for prosodic and phonetic function. On the left are MNI brain slices that show the candidate regions, aka the regions that were identified as significant via permutation testing, in blue, and the critical network, aka the single combination of candidate regions that minimizes prediction error, in green. On the right is an exploded MNI brain showing the ensemble network, or the sum of all combinations of regions that outperformed the control model. The color of individual brain regions is the frequency of an individual region within the ensemble. For example, let's take a look at this bright pink region in the second slice. This is Human Connectome Project Region 8C, a subdivision of Brodmann Area 8, which was present in 43% of networks that significantly outperformed the control. Let's zoom out now to the whole panel now that you know what you're looking at. The top row is a gray matter atlas, the Human Connectome Project Extended Atlas, and the bottom row is a white matter atlas, the AAL-CAT atlas, which ships with NESTAT. In general, prosodic function maps onto dorsal sensory motor cortex, dorsal and ventral motor cortex, so not as clean of a divide between VPCSA and DPCSA as we expect in the Beyond Broca model, the piriform cortex and the putamen, as well as corticospinal and cerebellar white matter pathways. The multitude of connections to deeper motor structures like the putamen and cerebellum makes me think this is due to the region's role in primary efferent control of the larynx. Piriform cortex is a bit of a head-scratcher, but this region is medial to auditory cortex and the temporal lobe and insula, so maybe this is reflecting connectivity to auditory areas as proposed in the original model. Pause the video now if you'd like to examine the prosodic results further. And here is a similar panel but for phonetic function. Phonetic function appears to map to ventral sensory motor and motor cortex, but there is still some dorsal involvement, so again, not a clear dichotomous split between VPCSA and DPCSA. Stroke data is messy after all, a lot of regions in the frontal and parietal opercula as well. What really stands out to me in these results is the white matter atlas. The one white matter pathway that is clearly the most frequent in the ensemble model is the arcuate fasciculus, a very classic language pathway linking canonical Wernicke's and Broca's. I believe this is indicative of the inferior parietal connections that support phonetic function. The arcuate also passes underneath the Sylvian parietal temporal, or SPT, junction, an important region for converting phonological representations into motor commands. Pause the video now if you'd like to examine the phonetic results further. So, to wrap up, we found separate pathways supporting prosodic and phonetic subscores of ASRS in a cohort with a large proportion of people with apraxia of speech. Prosodic function localized to central sensory and motor areas, connections to auditory and insular regions, as well as the cerebellum and subcortical motor nuclei. Phonetic function localized again to central sensory and motor areas, but with a more ventral distribution, as well as strong connections to the inferior parietal cortex. The dorsal ventral split between the two pathways of the beyond Broca model wasn't entirely clear here, but I still think these data support that split as, most conservatively, a heuristic. After all, this is only one study in a stroke cohort, and stroke can have a lot of individual variability. I'm personally still a believer in the dual motor coordination model. One aspect of motor coordination as it pertains to prosody and phonetics I would like to stress is the importance of these long-distance connections we identified. They really stood out to me in analysis, and moving forward, I would like to investigate the role that auditory/parietal cortex, as well as the cerebellum and insula, play in supporting prosodic and phonetic function. Lastly, as this is an aphasia conference, I think these results provide evidence that treating post-stroke AOS as a division into prosodic and phonetic subtypes could be useful in diagnosis and treatment, but more work is needed. Please let me know if you have thoughts on this or anything else in the poster. Lastly, here are the references and a QR code that has a bunch of links to relevant stuff from this project. If you are planning on going to AOA in person, this poster will be on display in the Horton Grand Hotel from 8 to 9:30 a.m. on Monday, October 27th as part of the Fluency, Speech Production, and Discourse session. I hope to see you there.